Professor Cremer is sharing his screen here and getting ready. I'd like to thank everyone again for tuning in this evening for our presentation here. Just a quick background of Dr. Cremer here. He is currently the Lloyd Huck Chair in Natural Science at Penn State University. He holds a dual appointment there in both chemistry as well as in biochemistry and molecular biology. Dr. Cremer received his bachelor's degree from the University of Wisconsin-Madison in 1990. He earned his PhD at UC Berkeley in 1996. He began his independent research career at Texas A&M, where he stayed for 14 years after being named Distinguished Professor. In 2012, he then moved his lab over to Penn State, where I got to interact with him a little bit before I graduated. So I'm very excited to have him here and hope everyone enjoys his talk here on supported lipid bilayers.
Thanks, Brad. And it's a pleasure to be here with you tonight and tell you a little bit about what we're doing in our laboratories here at Penn State. So the general title for the talk was The Behavior of Supported Lipid Membranes that I provided last year. And more specifically, I'll be talking about the work of a particular graduate student, Simu Sun, who had her PhD thesis, she just finished up, on a lipid molecule called PI₄, and I will introduce that to you shortly and tell you what she's actually accomplished. But since this is a talk mainly about analytical chemistry, I'll be introducing one new technique that she has, but also she did some beautiful spectroscopy work as part of this project. So what we work on in our laboratory typically involves understanding interactions and interfaces. And one of the most important interfaces from a biological or biomaterials point of view is a membrane that separates the inside of a cell from the outside of a cell, has a myriad of different functions and can be really thought about in many different ways. So from a chemical engineering point of view, this can be thought about as a liquid crystal. So any two lipids in a membrane, this is fluid, will change place with each other and do so in about the time scale of about 100 nanoseconds or so. So everything's free to move around in 2 dimensions. So I'm showing you here a biological entity with many binding sites on it. In fact, multivalency and bivalent binding will be the hallmark of Simu's work on PI₄. But if you take the entity I've shown here, you can come down, maybe bind once at the surface and then rearrange and bind multiple times due to the fact that it is 2-dimensionally fluid. The reason why this works out this way is because unlike, silanes on glass or thiols on gold, we don't have our chemistry, our surface chemistry, covalently bound to the surface in any way. There's a van der Waals interaction between this lipid membrane here and the underlying surface. That actually holds it to the underlying surface. These bilayers are made by a technique called vesicle fusion. Actually, you can use what's called Langmuir blodgetry and a number of other techniques. But what you typically have is what I'm showing you here, an underlying substrate that's typically glass. It usually is hydrated. So between the bilayer and the underlying surface are usually a few layers of water. And by a few, I mean 2 to 4 layers of water. So the thickness of these membranes is typically anywhere from something like 2 to maybe 4 layers of water thick. So maybe just the first hydration shell of the lipid head group and maybe a second, as well as that of the surface. These glass surfaces, we typically use borofloat glass, which is the main component would be silica. These are underwater, of course, because this is a biological environment that we're trying to mimic, and therefore they're negatively charged. So surface silanols, SiOH, tend to be deprotonated, or some of them tend to be deprotonated at neutral pH in the buffer conditions that we're working in. So you have a negatively charged surface that actually helps to orient and hydrate these head groups here. These head groups are zwitterionic. That's one of the forces that holds things to the underlying surface. So we have a 2-dimensionally mobile surface that we can use to make a biosensor or mimic from. And the chemistry that I'm going to employ these to use is going to actually involve this idea of having myriad components in the membrane. So what we're interested in studying with these membranes is to try to understand metal ion binding. It's been a theme in my lab for a very long time. Long time ago, Singer and Nichols argued that the lipid membrane was nothing but a fluid mosaic. What you had are proteins in the membrane, maybe you had sugar molecules attached to the membrane, cholesterol that I'm showing you here in yellow. Certainly things would come and bind to membranes, so maybe you'd have a metal ion coming down and binding to some alpha helical structure, or you might have metal ions translocate through some sort of a protein pore. But what really wasn't appreciated until the last decade or so is that the lipids themselves are not just a scaffold for the membrane, but they can actually play a direct part. And one of the first roles we were able to see was with this molecule PI₄ that we happen to employ. And what I'm showing you here is a metal ion binding site that will actually involve two different lipid molecules. Now, the reason why metal ions are going to bind is because we're going to have phosphate, which is going to be negatively charged and on these surfaces. So these lipid molecules are going to be highly phosphorylated, and the metals can come down and bind to two separate lipids. The understanding of how metal ions should come down and bind to surfaces should involve what's called Hofmeister chemistry. So metal ions, along with many, many other properties, obey what are called a Hofmeister series. This is a series that was found in the late 1880s by Franz Hofmeister. He first looked at proteins and their precipitation from solution. And if you take maybe the chloride salts of all of these metal ions, you'd find that they would follow the same regular series. If you put calcium chloride in solution, that might actually help solubilize the molecule, and things on the other hand would help precipitate from solution. Formation of micelles, turnover rates of enzymes. But if you were interested in anything from the pickling of cucumbers to how lightning storms occur, this actually follows a Hofmeister series. And you get the same series for the binding of phosphate in water in the form of a lipid that I'm going to show you, this PI₄, binding to these metal ions. Cesium will bind the weakest and calcium will bind the strongest. And the two ions that we'll be concerned about most in this particular presentation are magnesium ion on the one hand and calcium on the other. They sit right next to each other in this series. They bind the tightest of all of these Hofmeister series ions to the particular lipid of interest. And that binding constant only varies by a factor of 2. And yet the chemistry that I'm going to show you says that there's a great difference in their behavior. And so that was a real mystery to us and something that I want to share with you here tonight. So as I told you, I would tell you about this particular phospholipid, PI₄. So this is a phosphatidylinositol. A non-inositol ring is what's shown right here. So if I just have cyclohexane and it has OH groups substituted all the way around, then that's called inositol. Those OHs can be further phosphorylated, and here at the 1 position, at the 4 position, and at the 5 position, they're phosphorylated. If they're perphosphorylated, that's actually called phytate, and that's a common ingredient, for example, in cornstalk, in nutshells, and in many other things, along with the requisite metal cations to go along with it. But this inositol ring is also an important signaling agent on lipids. So when you attach it to diacylglycerol, so this is a glycerol backbone here, so 1 oxygen, 2 oxygens, the second oxygen is down here and the third oxygen is over here, form the backbone of this molecule. You acylate it here at the first position and the second position, and on this final position, position 3 here, you put the head group, this inositol ring. This is just about the most highly negatively charged lipid that you have in the body, to have this diacylglycerol happen to have this inositol attached to it. When it's PI₄, the biochemists, unfortunately, usually just ignore. They consider this first position here to be structural, just for attachment. They consider these two phosphates to be the business end of the molecule. But if I include also this backbone that has a negative charge, physiological pH, this molecule usually has one of these two phosphate groups being one deprotonation site, and the other one can usually be two deprotonation sites. If this is pKa of 6.7 and I happen to be working around 7.4, which is physiological pH, then I usually have a charge of something like -4 on this molecule. So it's a heavily negatively charged molecule, and you can imagine that it would attract metal ions, specifically magnesium and calcium ion. But this molecule performs just a ton of different jobs inside the membrane. It works as a second messenger, and I'll explain what a second messenger is. It is involved in enzyme activity. It's involved in cytoskeletal attachment, and I'll show you a little bit of a moiety about this. Endocytosis and exocytosis of various products into and out of the membrane are all trafficked basically based on this lipid. It's a very promiscuous lipid. Over 100 different proteins are known to bind to it, which is really quite amazing. And one I'll show you tonight is what's called the pH or Pleckstrin homology domain that actually also binds to this molecule. But I'm going to start by showing you its role as what is called a second messenger. So let me start by explaining that this sits inside of a cell. So what I'm showing you here in blue is the plasma membrane of a cell. This lipid PI₄, with its negatively charged head group, will be sitting on the inner leaflet of this membrane. It works as a second messenger. So what a second messenger basically means is it goes second. The primary messenger might be some chemical signal, and this chemical signal would be from the calcium pathway. It basically says up the calcium concentration inside the cell. So how does this work? A messenger comes along, the first messenger. It binds to either a G protein coupled receptor or receptor tyrosine kinase. In the case of binding to G protein coupled receptor, that releases G alpha. G alpha then comes and actually activates protein lipase C. This protein lipase C is a catalyst, and what it does is it takes this molecule PI₄, usually called PIP₂ by the biochemists, and it makes two products out of them. It makes what's called inorganic phosphate 3, which is just the inositol ring with the 3 phosphates, and diacylglycerol. So what one actually does with phospholipase C is you take, at this oxygen here, you hydrolyze the molecule, leaving this diacylglycerol behind and this IP3, this inorganic phosphate 3. So 1, 2, 3 phosphates is attached to it. When this species gets released, goes out into the cytosol, eventually comes down to the endoplasmic reticulum where there's a binding site. That binding site, when the IP3 binds to it, releases calcium, because calcium happens to be stored in the endoplasmic reticulum, releases calcium into the cytosol, and eventually this molecule can become phosphorylated, and that will turn off the signal. What I will show you is there's actually another pathway to turn off the signal. What was so curious to us and our collaborators when we started working with this particular membrane lipid is the fact that this process happens to occur in the presence of calcium at concentrations of about 100 nanomolar, pretty low, and magnesium at concentrations of about, a few millimolar, so about 4 orders of magnitude higher. Something like 1 or 2 millimolar of magnesium ion is inside the cell, 100 nanomolar or so of calcium is inside of the cell. There's more calcium outside of the cell. And in fact, there are actually membrane proteins that can transport it directly into the cell, as well as its storage here in the endoplasmic reticulum. Now, when you have this phospholipase C, it can actually hydrolyze this particular lipid, forming the IP3 and the DAG quite easily in the presence of a millimolar concentration of magnesium. But if you have a millimolar concentration of calcium around, this process will not go at all. It becomes completely shut down. And so there was no understanding when we approached this problem to try to figure out what was going on, despite the fact that it is well known in the literature that this particular binding site here, right at the bottom of the phospholipase C, called a Pleckstrin homology domain, will bind to this particular IP3 in the presence of no metal ions just fine. And in fact, there are well-known X-ray crystal structures, and these have existed for over a quarter century now, showing exactly residue for residue how this IP3 actually sits inside of this Pleckstrin homology domain. This is, remember, just a little piece of the phospholipase C. It binds to the IP3 with micromolar affinity, so that's pretty tight binding. And there's no evidence for any metal ion that sits in this site. So this is curious. Again, going back to the idea that when you have a fluid mosaic model for a membrane, usually one imagines that if something, in this particular case, hydrolysis reaction, works perfectly well in the presence of magnesium ions, so this IP3 and DAG will split apart, but won't work at all in the presence of calcium. The first thing that you would turn to try to understand a problem like this is the X-ray crystal structure. You might look for a particular binding site for one metal ion or another that might indicate why magnesium will make this process work and calcium won't. But there is no binding site. In fact, only the phosphatidylinositol lipid will bind, and the metal ion will not bind to this binding site. So we worked in a team on this project. We had collaborators over at the Max Planck Institution in Dresden, Germany, led by a collaborator of mine named Uno Kaskun, and he was doing old-fashioned 1950s analytical chemistry assays or bioanalytical assays called a flotation assay to try to understand this particular system. He would take a test tube and he would put lipid vesicles in it. These vesicles would contain a regular lipid called phosphatidylcholine. This is the most common lipid present in your body, and he would add about 5% of this PI45P2 to it. This PI45P2 will bind to that pH domain, that Pleckstrin homology domain, the same one, right, that is active in this phospholipase C, and he would add in this pH domain that's being shown here in orange. The vesicles are basically being shown here as spherical shapes called large unilamellar vesicles, about 100 nanometers or so across. He'd fill up the test tube with solution with this pH domain, and then he'd spin it down with a centrifuge. It's called a flotation assay because vesicles will float. If you have unbound proteins, they'll actually spin to the bottom once you centrifuge this. But of course, those that are actually bound to these lipid vesicles, they'll come to the top for a ride. So one can actually look now, you can use fluorescence. Back in the day, they used to actually use a lot of radiation and a Geiger counter to see where their proteins were bound. But if you just take a fluorophore and put it on your pH domain, you can see the fraction that are on the top and the fraction that are on the bottom after you centrifuge. So that's what I'm showing you over here on the right. So a top fraction, there's actually nothing left in the middle after you centrifuge. And although this picture really isn't drawn to scale, you see two proteins on the top and two proteins on the bottom. In the particular assay that was run here, most of the proteins ended up at the bottom here. But in the presence of PI45P2, 5 mol% in these vesicles, you get a fraction that binds to the top and comes to the top. If we then add 1 millimolar magnesium to the solution, so in the same solution that you put into this test tube here, that you have a millimolar magnesium, the assay works just as well. The PI45P2 binds to these pH domains, And some fraction of these shown here in green float to the top, 1 millimolar magnesium. If we take, instead of a millimolar magnesium chloride, if you take a millimolar calcium chloride, run the otherwise exact same assay again, what you find is that there's no top fraction. You can see the bottom fraction here. If you compare the green to the red, you have all of your proteins end up being centrifuged to the bottom. None are with the lipid vesicles here on the top. You can run one final assay, and that is with just PC vesicles with no PI₄. So if you don't have this particular lipid that binds to this pH domain, then all of the pH domains will go to the bottom. So this is rather curious. I've shown you an X-ray crystal structure. There's no binding site on the protein for either calcium or magnesium, yet when you actually go run a simple ligand receptor binder between PI₄ and these pH or Pleckstrin homology domains, you get an enormous difference. They'll bind just fine in the presence of magnesium, but they won't bind at all in the presence of calcium. So the first thing we chose to do was run a rather modern assay, one that we've invented in our laboratory in the last 10 years. So we look for interfacial potential changes using potential sensitive dye. And we can actually monitor the binding of calcium to these phosphates here, because what they end up doing is they end up charging the membrane up positive when they come down and bind to the surface. And so as a fraction of divalent metal cation that we plot here on the x-axis versus the fluorescence intensity change that actually causes the fluorescence to drop, you can see, as I've advertised before, calcium and magnesium are very similar. So if you remember back, I showed you this Hofmeister series at the beginning of the talk, and these two ions sit right next to each other. As a consequence, you wouldn't expect much of a difference in binding, and indeed, the binding is almost identical. So within a factor of 2, the equilibrium dissociation constant for magnesium ion to these phosphates is about 1.2 millimolar. The equilibrium dissociation for calcium is only about a factor of 2 or so tighter at about 0.6 millimolar. So these interaction affinities are just about the same. So if you're not binding to phosphate much differently, then what is causing this huge difference between what you see here as a bound fraction in the presence of magnesium ion and no bound fraction in the presence of calcium ion? A real mystery when we started out. So we chose to use spectroscopy to directly address this problem. And the spectroscopy that we chose to use is a nonlinear optical spectroscopy that we employ in our laboratory called infrared visible sum frequency generation. This is a surface-specific vibrational technique that combines an infrared laser beam. We happen to be doing this with picosecond lights, so we use a neodymium YAG laser. This is a doubled YAG frequency that we employ. These are on the order of about 17 in our particular case picosecond. Should be about the duration of the photons that are hitting the surface. And we also generate infrared light that also is on the order of about 17 picoseconds. We shine them colloquially on a surface, in this case of a Langmuir trough. So a Langmuir trough is just a big bucket of water where we're going to put a lipid monolayer on top. So if I blow up a picture of that, here's a lipid monolayer instead of a bilayer. This lipid monolayer will be PI₄. The alkyl chains will stick up into the air. The head groups, this phosphatidylinositol will stick down into the solution. And if we have cations in the solution, they should be able to come up and bind. And so this particular technique, infrared visible sum frequency generation, sometimes just called SFG, because you generate a photon at the sum of these two frequencies, IR plus visible, obeys selection rules that are a combination of infrared and Raman selection rules. And this turns out this combination of infrared and Raman selection rules make the technique surface specific. You have to break inversion symmetry in order for you to see something that happens at the interface, but not in the bulk solution, which is isotropic. So we employed this technique to look at this monolayer interface of PI₄. We did it in the case of calcium and magnesium chloride, as well as the first case that I'll show you here, just pure buffer. So we have 10 millimolar of tris buffer that we happen to put in solution. We keep this at pH 7.4. And now what we're going to do is monitor the solution by SFG, or monitor the surface, and you see one peak that's down here between 950 and 1000. This is a breathing mode of the inositol ring, and another peak just above it. This is a phosphate symmetric stretch. I'll come to this one in a second, but let's look at this lower frequency mode. This ring breathing mode is just the breathing mode of the inositol ring, and it's a pretty small or modest peak. But what you can see happens when I add a millimolar magnesium chloride to this solution, it picks up a bit, but it picks up a whole lot more when I add calcium chloride to the solution. So if you think about a monolayer like this, the number of lipids that we have here at the interface are basically the same. So what metal ions can do when they come and bind is they can reorient the moieties to which they bind, and they can also order them at the surface. And SFG is terrifically good at seeing things that are ordered. And what's happening here is that you're getting a modest degree of reorientation and ordering in the case of magnesium, but it's a whole lot better in the case of calcium. Calcium is doing what magnesium cannot, it seems to be. It's implying or impressing a great deal of order on this membrane. And a clue as to what's happening comes from this phosphate peak right next to it. So what you have here is a peak that starts out just below about 1100 wave numbers here in this SFG spectrum, and that's from the symmetric stretch of these phosphates here on the 4 and 5 position. So the phosphoryl stretch here and the phosphoryl stretch here show up, and they show up at 1100 wave numbers, and that's only underwater when they're well hydrated. If you would have this out in air, this same phosphate group would end up stretching that phosphoryl P double bond O at something like 1200 wave numbers. The reason why it's lower frequency is the water hydrogen bonding to it actually leads to a little bit of charge transfer from the lone pair on the phosphate to the sigma star orbital of those waters. And that actually weakens the bond just a bit down to 1100. So the interesting thing that you're seeing here is you're seeing a blue shift of this peak in addition to its picking up here when we start adding these divalent cations to the solution. The metal ions, when they displace the water and bind to the phosphate moiety, eliminate the ability to do that charge transfer from those lone pairs on the carbonyl, on that phosphoryl oxygen onto the sigma star orbital of water. So the metal ion comes down and blocks, and that actually causes a blue shift up back towards the gas phase frequency here, something like 1124, 1125 wave numbers or so. And you can see that the shift occurs modestly in the case of magnesium, where this peak is still the higher one, but you clearly see in the case of calcium that calcium has more binding that is displacing. And you might say, huh, didn't I just tell you that the binding constant for these two metal ions was almost identical? And in fact, I can do this at many different concentrations. This is 1 millimolar. You get almost the same result if I do this at saturation with 10 millimolar of calcium or magnesium chloride. The difference that you're seeing is a result of the type of binding. Magnesium is the smaller ion and really likes to have exactly 6 waters in its hydration shell around it. And when it comes and binds, if my first fist is phosphate and my second fist is the magnesium ion, it binds solvent shared. When you buy magnesium chloride from, let's say, Aldrich, you never buy just magnesium chloride, you buy the hexahydrate because it's very hard, and magnesium is pretty hygroscopic, likes to hold on to those waters. Same thing in solution. You can think of it in a biological or aqueous solution as being much larger than calcium because its 6 water molecules come along for the ride with it. And that's exactly the evidence that you see here. This peak sits here. This first peak is evidence that much of the magnesium that's binding is binding in what's called a solvent shared configuration. Not so for calcium. It's a little bigger ion. Instead of having 6 waters that are well hydrogen bonded, you have a 7th water around the calcium. And so since it's a little bigger ion, so the charge is more dispersed on this, and you have to accommodate 7 waters, those waters are bound much more loosely. In fact, water will exchange from calcium with a neighboring water in about a 5 nanosecond time scale versus it's more like 5 microseconds, 1,000 times longer for a water molecule to exchange out of the first hydration cell of a magnesium ion that's hydrated in an aqueous solution. And so this contact ion pairing that you're seeing here versus much less contact ion pairing that you're seeing in the case of the magnesium ion is decisive. So calcium is directly bound with a contact ion pair. More of the magnesium is in a solvent shared ion configuration. And that's leading to much better ordering here of this inositol ring. If we look further up, this is the carbonyl ring. So now what we're looking at is further into the backbone that we're seeing that this carbonyl stretch is organized. Now I'm going to show you way up here on the end of the molecule as well as the water structure. So this is the CH region. Calcium orders this lipid such that all the way at the end, these methyl groups at the end, which are giving rise to this peak, are better ordered. In the presence of buffer without divalent metal ion, you see these are the 2 peaks for water. The water comes down modestly in the case of magnesium, but because calcium actually displaces more water by binding directly, this peak here, this very broad water spectrum, is actually a bit lower. So we have evidence for direct binding. We have evidence for reorienting, which is what's causing this huge rise in the signal of this inositol ring. So we turn to our third collaborator in this project, Pavel Jungwirth, who works at the Czech Academy of Sciences and does molecular dynamics simulations. And his molecular dynamics simulations of these membranes, this is a membrane here with 128 lipids, so 64 on the top, 64 on the bottom. These are all atom molecular dynamics simulations. And of course, he's also got this PI₄ in it. On the left-hand side, this is without any calcium ions. But when you add calcium ions in solutions, you get many of these snapshots that look like the one over here on the right, where you see calcium ions are actually bridging 2 head groups together. And this is something that the SFG data shows you that the magnesium ions will not do. The magnesium ions can be thought of maybe as a large beach ball, right? And so although they bind, they're in this very large ion configuration trying to hold 2 together that really doesn't work so well because you shed the hydration shells and the calcium will bind tightly. You actually glue phosphate, calcium, and then another phosphate. This will actually cause these rings to bend over towards the membrane. They take on an angle of about 37 degrees with respect to the plane of the membrane. And that orientation change is exactly what you're seeing causing this peak to pick up. So SFG is extraordinarily good at seeing ordering. If all of the PI₄ is now ordered such that it has an angle of 37 degrees, that's what's causing this peak to shoot up so much. Since magnesium really doesn't do this as well, you don't see it pick up as much. And this was reflected in the carbonyl stretch, reflected all the way to the CHs, this ordering of the membrane and this reordering of the lipid all the way to the other side. A cartoon of what's actually going on in this process is shown here. So this Pleckstrin homology domain, this part of this phospholipase C called this pH domain, will bind to phosphatidylinositol with this micromolar binding constant. But when I have calcium present in the cell, this causes contact ion pairing and the dimerization of these lipids. They bend over towards the surface, and that inositol ring is no longer available to bind to these pH domains. And so basically you shut off the binding process. So if we go back and look at the molecular cell biology that's involved in this, I showed you this calcium signaling pathway. So these were these first messengers that were causing the signaling, and you had this hydrolysis process. Interestingly, once you start releasing calcium into the cell, That calcium, of course, can start binding to these lipid molecules, and that's one more means, a redundant means of actually switching this process off. So at first you were having this phospholipase C, it was hydrolyzing PI45P2 to IP3 and DAG, and once the calcium was released, that caused these lipids to bend over towards the membrane, and then it could actually no longer continue this process. So it was working as an off switch in the biological process. So this is something that will really only work in the presence of calcium, not in the presence of magnesium. And so what you're seeing is, unlike the fluid mosaic model of the 1970s, which really thought that only the proteins were doing the work of structure function within the membrane, that the lipids themselves are not just a scaffold for the membrane, but in the case of IP3, it's really able to function not just as a second messenger, but as an off switch that stops the enzymatic catalysis that was occurring. So that is an example of one vignette of this signaling pathway. And so the second vignette that I'm going to tell you about tonight from Simu Sun's work is cytoskeletal attachment. So this lipid is the only known lipid that actually holds a plasma membrane to the underlying cytoskeleton. So I'll show you a cartoon of that. So there are usually actin filaments inside a cell, and these actin filaments sit right under the plasma membrane that's being shown here as a line. And it's been known by the biochemists that these PI45P2 molecules will become immobilized on these actin filaments. And the biochemists guessed that this was probably patches of positively charged positively charged proteins under here, arginine and lysine residues on these actin filaments that were holding these particular highly negatively charged lipids in place. There are no other lipids that actually attach the plasma membrane to the underlying cytoskeleton. And if you delete these PI45P2 from the plasma membrane, you can actually watch the whole membrane detach itself or delaminate it from its underlying cytoskeleton. It's quite a remarkable thing when you look at it under a microscope, because the plasma membrane actually becomes floppy at that point. So it's actually directly involved in making of the structure of the membrane. So the reason why we were interested in this came from this first project that I just showed you on this binding of these pH domains that will occur in the presence of magnesium, but not in the presence of calcium. When we did these experiments, we noticed some other things that were quite curious, and I'm going to tell you a little bit about that here. So I'll put a dye molecule in these membranes. This happens to be Texas red. This is a chromophore that's basically a tied up rhodamine. So these nitrogens here are tied up in these ring structures. If you hit this with green light, it will fluoresce in the red. We're going to put this in our membranes in 1 part in 200. So 199 parts will just be this regular POPC lipid. So it has a positive charge on a choline, a negative charge on a phosphate, and I'm going to have this dye molecule. And so at first here, I'm showing you with no PI45P2 in the membrane. What you have in this before case here, you're looking top down. So I'm holding a membrane here. I'm looking at a microscope where I'm shining a laser light green, and I'm looking for the fluorescence, and I'm looking for a bulb. And so this is basically just uniform fluorescence intensity. I now will hit this with a laser beam, creating a little spot. The spot's about 10 micron or so across. And because these lipids are fluid, that what I can do, because I've photo bleached a spot, basically there's a little bit of oxygen in the water, and that oxygen will destroy what's called the xanthine ring here. That's these 3 conjugated rings in the middle of this molecule, which are actually causing its fluorescence process. So in the presence of a little bit of oxygen in your water, if you hit this with light, the molecule goes into its excited state, will bind the oxygen, and it will destroy the molecule rather than fluorescing. So that's what's creating this dark spot. So these molecules, a small portion of them are destroyed, but because this membrane is fluid, you're going to see other fluorophores come from the outside to this spot. And so you start to see this recover with time. That's evidence that it's actually a two-dimensionally fluid film that's on my surface here that we're looking. And finally, it's completely recovered because you have such a large pool outside of this. And you can watch what we called fluorescence recovery after photobleaching. So at time t equals 0, we basically photobleached a spot, and we can watch this recover as a function of time. Those are just these points that were going out here as a function of seconds out to about 1200 seconds when you're watching this. Over the course of something like 20 minutes, you're watching a recovery. Here we fit this to a single exponential. That's that red line here. And that single exponential fit is to just this equation here, this decaying exponential. This term is what people are usually interested. This B is the characteristic time constant for how fast these recover. And the recovery time for the experts in the audience in lipid membranes usually refer to something on the order of a few microns squared per second for the diffusion constants of lipids or proteins in membrane. Those diffusion constants correspond to about, the viscosity of molasses or honey. So the membrane is actually moving quite a bit slower than the water molecules around it. As I told you at the beginning, you're changing the place of 2 lipids on about 100 nanosecond timescale or so. But what we're really interested in is the fraction recovery here, A. So when we've photobleached a spot, you can see that this is recovering to about 98 or 99%, right, over long periods of time after you wait a long, long time. And those are these Texas red molecules. We can do this differently. Instead of looking at this A constant for Texas red, we can look at it for PI₄. But before I show you that, let me just show you what should be happening here. You have these lipid molecules, they're mobile, so they move back and forth. But what we found here with our PI₄ containing membranes in that last set of experiments is that it appears as if like some of these are being held, maybe even by the surface that I'm going to show you here with an assay here in a second, so that you get a very low mobile fraction. You don't get 99% of these to be fluid. In fact, if we attach the fluorophore not as Texas red to a head group, but now I'm going to attach what's called a Bodippy fluorophore. I'm going to put it on the tail of one of these phosphatidylinositides. So not grown to scale. The head group here looks bigger than the one down here, but they are in fact the same head group. So I'm going to put PI₄ in the membrane here in the form of what some of what's called commercially that you can buy this as top floor. Top floor is to just put this Bodippy here in the tail. And if this molecule becomes immobile in this assay, then you will see that this fluorophore will stop moving. And so this recovery process that I showed you back here shouldn't occur. And in fact, if I show you the fraction that recovers on a chart like this, what I showed you with POPC membranes where we doped in 1 part in 200 was Texas red, we could do one of these fluorescence recovery after photobleaching or FRAP curves. We could do it initially within the first 20 minutes or so, and you get 98% or 99% recovery. If we run this 1200 minutes, so something like 20 hours later, you still get 98, 99% recovery. But if you looked at these PI₄, where now I put this fluorophore here on the tail, these are terrible, right? I'm only getting 60% recovery here within the first few minutes after forming the membrane. But if I do this FRAP assay a little bit later, what I'm finding is the next day, now I've only got about 40% of it mobile. So you had a poor mobile fraction, and this is getting worse with time. So when I had this initial condition here, I've got some of these fluorophores. It's not very good compared to just a regular POPC membrane. So the next thing we figured we'd try is forget this bilayer, let's make a new assay and let's run this as a monolayer. So on a hydrocarbon surface here, what we can do is we can put just a monolayer on top. And now initially we have something like 84, 85% that's mobile instead of 60% that's mobile down here. And this doesn't change as a function of time. So these particular monolayers are not nearly as nice as the bilayers. Even if I had shown you POPC with a little bit of Texas red in it, it's no longer 100% mobile or 99% mobile. It would be down here too. These lipid monolayers are not as high of quality, simply usually because the alkyl chains, which are surface silanols, are usually a bit more disordered on the surface, but they're still pretty good, 85%, and this doesn't change. So this is one hint that it must be this underlying surface that's making these immobile. So the next thing that we tried to do is, let's just make a bilayer again, just like we had here, but let's take this pH domain and we'll try to tie up this head group here with this very large protein, or this piece of a protein that's binding micromolar. See if we can hold this and stop it from flipping to the other side. And sure enough, it works. So if I run this assay in the presence of about 100 micromolar pH domain, then I start out at about 63, 64% mobile fraction, and 20 hours later, that mobile fraction stays identically the same. So it seems to be that it's this lower leaflet that's responsible for this problem here of becoming immobile. One more experiment with this pH domain that you can imagine doing as a check is you can make a binding constant measurement, and you can show that the binding constant of pH domains to these underlying surfaces that I'm showing you here. So changing the fraction of pH domain that I put in, make a nice titration curve, looks like a Langmuir isotherm, looks like the binding constant or the equilibrium dissociation constant is something like 600 nanomolar. But if I call this fraction of protein that I get at saturation when I have 10 micromolar protein, 1 here at the end of this assay, and I now wait 6 hours and run the assay again, now I don't get nearly as much protein on the surface. When I go to make the binding constant again, the equilibrium dissociation constant, that's just about identically the same, but now what you're seeing is that you're only able to get about 60% as much protein. You're losing about 40% of these lipid molecules, which seems to be similar to what's happening here going on, that we're getting a decrease. So our hypothesis is what's causing this immobilization process to occur is that you have some fraction of the PI45P2 in the upper leaflet, and those remain mobile at the beginning of an experiment. But every time they flip to the lower leaflet, these become immobile. So our hypothesis in this process is that these are flipping from the top leaflet to the lower leaflet. So initially we're starting with about 60% mobile, so 3 out of 5 are in the upper leaflet. When we initially make these membranes, that makes sense because these are negatively charged lipids. The glass surface is also negatively charged. So when you initially make the bilayer on electrostatic grounds, you might expect these to repel. But somehow, over time, some of the lipids are able to actually flip to the bottom and actually saturate this glass surface with these PI45P2 molecules. And so to prove this, so I've given you what I would really call anecdotal evidence that this is true. The graduate student, Simu Sun, who did this assay, created a fantastic new analytical assay. So for the first time with supported bilayers, you really want to be able to analyze separately what's in this upper leaflet from this lower leaflet. And her new assay is called an unzipping assay. So if you were to just unzip this lipid bilayer into 2 halves, in the upper leaflet and the lower leaflet, you could actually look because you have fluorophores right on these tails. So you would see how much fluorescence was in one membrane versus the other. So if you could separate these two, that would actually be an ideal way of actually telling them apart. And sort of the inspiration from this came from microtoming experiments that go back to the 1960s and 70s. Even with a cell membrane, when you microtome things and split them into pieces, you can sometimes see that the bilayer will actually split apart at the alkyl chain at the hydrocarbon region because those are only being held together by van der Waals forces. So Simu Sun argued that she should be able to do the same thing with our assay. So you start with a lipid bilayer on a planar solid support. You put another piece of glass on top, so one piece on the bottom, one piece on top. She then put a weight on top of this to actually try to push out as much water as you could. And when you do that, she then vacuum dried this and split this apart. They were held together by pretty good adhesion forces. So you use tweezers to pull these apart. And you can do a number of different assays, one of which is atomic force microscopy. And you can quite quickly show that you have monolayers on each piece of glass that's left behind. On the lower part and on the upper part, you have a monolayer. And here is the monolayer by tapping mode AFM. And here's a scratch that we intentionally made. This basically just rubbed away the bilayer in this little portion here. And then there's, monolayer in this little portion here. And there's monolayer here on the other side. So if I take a line profile across this, what you can see is that this lipid monolayer here is about 2 nanometers high. That's exactly the right height that a monolayer should be. The total bilayer is about 4.5 nanometers. So half of that would indeed be exactly what we're getting out of here. Moreover, you can show that in humid air, these are fluids. So you really have lipid monolayers that are being able to be split apart by these assays. We're right now trying to actually go back and figure out the underlying mechanism for how this particular assay works, which we'll have out in a paper soon. But one of the first things we did, one of the first things Simu-san wanted to do once you made this assay is there were long running questions in the literature. If you finally have a new analytical assay that can tell you what's in the lower leaflet versus what's in the upper leaflet, one of the burning questions in the literature is, gee, if you have a neutral lipid, so if I take a tail labeled BODIPI, that what you have is a positive and negative charge. This is Switter ionic. One would think that its distribution between these two leaflets might be different than this Texas red DHPE, where you have one positive charge here, but one on the sulfonate here and one on the phosphate here, that you have an overall net negative charge, that if you have a negatively charged lipid, that more of it will end up in the upper leaflet. And that's because the surface is also negatively charged and these lipids will be repelled from the underlying surface. So not in this case, but in this case. So the first thing to test our handy dandy new assay is to try this BODIPI lipid, this uncharged lipid first. So she did her unzipping assay, looked at the fluorescently tail labeled lipids, and you can see that the fluorescence in the lower leaflet and the upper leaflet was about 49% to 51, very close to 50-50. But if you had Texas red, this looks more like 40% in the lower leaflet and 60% in the upper leaflet. Just exactly what people had been guessing at for years, that if you have charged moieties, that they will unevenly distribute through the two membranes. In fact, you can start adding salt and start to see how these might actually change their distribution if you try to screen the surface from these charges. So this assay really works, can actually tell the difference between an uncharged and a charged lipid. So immediately she moved to looking at her PI₄. And the first thing she did was she split apart her assay, and you can clearly see that there's much less fluorescence from the one leaflet than from the other. I would note here that these marks here she made intentionally. These were just scratch marks, so that she could actually tell that this location here was the same location that she had here later. This point here was actually, you can see it's very faint, but it's the same point here. These are defects that you have in the membrane when you do this. So same area that was in contact with one another, but 40% or so of the fluorescence in the bottom, 60% in the top. We can quantify that. It's about 63% in the top, 43% in the bottom. So let's wait 200 minutes and then do the unzipping assay. And then they're about even, about 50-50. And if we wait overnight again, so 20 hours or so later, now you can see that it's flipped, that more of the fluorescence is in the bottom than in the top. And so the immediate question that you might ask is, oh, let's compare this back to the FRAP assay, right? Where we initially had 60% mobile and it went to 40% mobile. And so that's shown on the graph here. So what's in the top leaflet and what's mobile, so what's in the top leaflet is shown in this light blue and what's mobile is shown in yellow. These correspond at 0 minutes. They correspond light blue to yellow at 200 minutes and light blue to yellow 20 hours later the next day. On the other hand, the immobile fraction, which is what's in the lower leaflet here by this unzipping assay, and the immobile fraction, which is this orange, so dark blue and orange, these two are correlated at 200 minutes and correlated 20 hours later as well. So one seems to be tracking the other. We seem to now have proof that what sits in the lower leaflet is exactly what's being immobilized. And somehow we're going from 60% fraction initially to, in the upper leaflet, to only 40% fraction in the upper leaflet later. So one could wonder, how are these lipids getting from the top leaflet to the lower leaflet? And so one thing that would occur to you is that they would be moving around defects. These things are highly negatively charged. It would be hard to believe that they would go through the middle of the membrane. But if there are these defect sites, we should be able to put protein down on the surface like bovine serum albumin. And since this inositol ring sits out of this membrane with these phosphate moieties on it, that should stop it from going around. And indeed it does. So if we run this assay without putting some BSA down on the surface to block these defect sites, then 60% initially mobile, 40% after 20 hours. But if we backfill this, now we start out with 60%, we're nearly at 60% even 20 hours later. So this really works. The last assay that I will show you is, I finished the talk, why are these things becoming immobilized on the surface in the first place? And the last assay I'm going to show you to try to explain this is this deuterium isotope effect assay. So what I've been showing you in the second half of the talk are these FRAP curves. The black one here shows that initially at 0 hours you run FRAP and you get about a 60% mobile fraction. So 1,000 seconds later, about 60% is recovered. And that if you run this at 20 hours, and that's this red curve here, only about 40% or so is mobile. If you run this same exact assay with the same mole percentage of PI₄ in the membrane, and you simply do this in D2O in your buffer, make it out of D2O instead of H2O, what you can see is just over 20% of the lipids are mobile at the initial time, and 20 hours later, that hasn't changed. You basically saturated the surface and no more PI₄ can possibly come down, even if you were to wait longer. And so this is a huge isotope effect. You're immobilizing much more of this particular lipid on the surface. So that seems to indicate that hydrogen bonding is playing an enormous role for immobilizing this particular lipid, as opposed to electrostatics. Remember, the surface is negatively charged. Some of these surface silanols are deprotonated, and certainly PI₄ is highly negatively charged. But unlike many other binding events, phosphate loves to hydrogen bond, and it's hydrogen bonding. It hydrogen bonds better to silanols than it does to water, also to proteins. Many proteins like serine residues, for example, hydrogen bonds much better to water. So you can imagine a hydrogen bond here. If you were to switch this to deuterium, that's been known for 50 years, from Harold Sharaga going back to this paper in the Journal of Physical Chemistry, that deuterium actually has a shorter bond length because the bond sits a little further down in this well. It's actually a bit of a stronger bond because it's this shorter bond length, so that bond tightens up. It's about 5% stronger. And so if you make these bonds about 5% stronger, and if they're multivalent, so let's say we have a couple of these, and in fact, you don't have to just be multivalent to one phosphate, you can imagine binding to both of these here, that this is likely to be causing this huge difference. And in fact, you can run a little model where you assume a Boltzmann distribution with a 5% difference in binding constant, and that will exactly explain this difference that I'm showing you here, just this switch from hydrogen to deuterium. So you might wonder, therefore, if you do this on a protein layer, what happens? And so this was initially an inorganic surface, glass, that we ran this assay on. But now, if we put a protein layer down on the surface, you see that you start out about 70% mobile for the PI₄ in the bilayer when it's on this protein cushion. And that number drops with time because, of course, you have hydrogen bond donors also from the protein cushion. So that goes down and it saturates. So this seems to be a generic effect. I can have protein layers. We've done this with polydimethylsiloxane. We've done this with glass. We've done this with various types of oxides. This seems to be a very general phenomenon, that what is immobilizing is not the negative charge on the lipid here to the glass, because of course the glass is also negatively charged, but it is the ability to hydrogen bond. And we would postulate that this must be exactly what's also happening here in vivo. These actin filaments are also negatively charged on net, although they do have some positively charged residues. And I don't doubt that if you have arginine or lysine, that could also help. But the main factor here to immobilize these on the surface must be the hydrogen bonding of the PI₄, with this big ring that sticks out, that hydrogen bonds to the underlying surface and becomes immobile. So with that, I'll end with these two papers from Simu Sun that I've shown you. So our group no longer takes our regular pictures. This is just a virtual picture of us in a Zoom meeting. Simu Sun graduated last year, so I'll show you her in an old picture from when she was still in the group. So there she is here in the back. She did all this work, and this work was funded largely by the National Science Foundation. And it's been a pleasure to come here this evening and to tell you a little bit about our work, and I'd be happy to answer any questions you have.
Thank you, Dr. Cremer, for your talk there. Is there any questions?
I'm happy to answer anything.
Okay, well, it looks like clearly you just explained everything so well. So again, I want to thank you for taking the time out of your busy schedule to present with us here today. On behalf of SACP, we really appreciate you coming to the meeting this evening.
Happy to do it.